اشتراک. صباح الخیر ممنون. شلونك؟ دكتور شلونكم؟ شلونكم؟ چطوری؟ چطوری means how are you in Persian? I'm fine, thank you. Oh, دیگه نمیان نه؟ باش. This lecture is about immunologic tolerance and autoimmunity. I told about this lecture yesterday a little, but today I'm going to talk about the mechanisms of immunologic tolerance. Understanding the mechanisms is very important in order to use them for treatment of autoimmune diseases. So tell me about immunologic tolerance. What do you know about it? What do you remember from yesterday's lecture?
Immunology intolerance maintained self-antigen in two locations, central tolerance, diaphragm tolerance, central primary organ, thymus gland and the bone marrow, and the diaphragm secondary organ. The central tolerance three part. One part B cells.
Thank you. That's right. He's talking about the mechanisms, peripheral tolerance, central tolerance, the location of them. Yes, thank you. That's right, all of them. I ask you to tell about the aim of the tolerance, not the exact mechanism.
Distinguish between self and non-self.
Distinguish between self and non-self.
Benefit bacteria and non-benefit bacteria.
Distinguishing between non-pathogenic microorganism and so why tolerance or immunologic tolerance is important?
Because if there is no tolerant immune system, we're going to attack ourselves. So
when there is no tolerance, you will have attack or invasion of self-cells or self-antigens by the immune system, which is called autoimmunity. We have several autoimmune diseases, and all of them are results of failure of immunologic tolerance. And today I'm going to talk about the immunologic tolerance and then mechanisms of autoimmunity, or whenever we have autoimmunity, indeed we have failure of tolerance mechanisms. So throughout the evolution, immune system tries to discriminate between self and non-self, and tries to not to attack not self-antigen, but induce a strong immune response, cellular, humoral, innate, adaptive, against non-self or pathogenic antigen. So I'm going to talk about how immune response maintain unresponsiveness, the mechanisms behind it to self-antigens, and then I'll talk about the factors
genetic factor.
Many factors that contribute to the loss of self-tolerance, which can lead to autoimmunity. And yesterday I talked about this part, unresponsiveness to commensal or no problem, it's the screen. Commensal bacteria, I talked about it. So, immune system, it's called immunologic tolerance. When we say we have intolerance or immunologic tolerance, we do not mean that inside the body we do not have self-reactive clones. Do you know what the meaning of clone?
Yes.
Lymphocyte clone?
Yes.
Could you please tell me?
It's antigen.
Clone of lymphocyte in one sentence, please.
It's a specific antigen make a population of its function. It's many T cells like it.
B cell that is specific to an isotope.
Isotype.
as a top.
Specific receptor for specific epitope.
Yes. A specific receptors against a specific. Does it mean clone? We have TCR, BCR antibody against epitope. Does it mean clone? What's the meaning of clone? Lymphocyte clone.
Population of some cells against similar
A population of similar cells that are producing antibody or they have TCRs against same epitope. A population of lymphocytes that all of them are same and all of them have receptor against one epitope, one kind of epitope. So when we say that we have immunologic tolerance, we do not mean that there is no self-reactive clone. As I said, immune system tries to produce billions of clones without exposure to all epitopes in the environment. It's not possible for immune system throughout the development of immune system, it's not possible for immune system to be exposed to all possible epitopes in the environment. Get it? It's impossible. We have billions of epitopes in the environment, but immune system should be able to respond all of them. But T cells and B cells are developed in the body without exposure to all of them. So without exposure to epitopes in the environment, immune system tries to develop billions of clones by chance that each clone will recognize its own epitope in the environment. So production of clones inside the bone marrow and thymus are indeed without exposure to foreign antigens. It's very important. For example, we have anti-HIV clone inside the body, but whenever we do not have HIV infection, the clone won't become activated. But we have the clone without exposure to HIV. Okay, we have clones of clones against COVID antigens. It doesn't matter if we have infected with COVID or not. So we have billions of clones against billions of epitopes inside the body and immune system. And besides, there are some clones against self-antigen. It's very important. Immune system cannot exclude, please, immune system cannot exclude all self-reactive clones during the development of the cell. It's not possible. So already we have self-reactive clone inside the body. All of us have self-reactive clones inside the body, but they do not react to the self-antigen in normal condition, unless we have autoimmunity. So inside the body, we have self-reactive clone. These concepts that I'm talking are very important. So we have self-reactive clones inside the body. After exposure, for example, in multiple sclerosis, MS, and autoimmune diseases that immune system attack nerve cells, all of us have anti-myelin clone inside the body. All of us. Anti-myelin, the sheet of the neurons. We have anti-myelin clone inside the body. But these clones are controlled in normal condition. They are not allowed to respond to neural cells. They are not, for example, other clones, we have clones against Langerhans island of pancreas, but they are not allowed to become activated. They are controlled constantly. So immunologic tolerance tries to control. First of all, it tries to eliminate some self-reactive. First of all, immunologic tolerance, there are some mechanisms that tries to eliminate self-reactive clones, but it's not possible to eliminate all self-reactive clones. We have some other self-reactive clones throughout the body that other mechanisms should control them, should suppress them, should prevent their activation. And we have self-reactive clone, and they have access to the immune system as well. So antigens are not excluded from immune system. We have both clones and antigens together inside the body, but we do not have reactivity. It doesn't mean that we do not have clone or we do not have access of antigen to the clones. We have both of them. We have self-reactive clone, we have access of antigen to the clone, but there is no reactivity. So both of them, antigens, I mean self-antigens, have access to immune system, and on the immune system we have self-reactive. But there is no active reactivity, there is no immune response, there is no activation of T cells, B cells, or other cells. Maybe T cells and B cells. So some mechanisms should prevent immune response to self-antigen, as we know that we have self-reactive and they have access to self-antigens. And some active mechanisms that constantly they are active inside the body to suppress the self-reactive clones. And if the mechanisms fail, they will have autoimmunity. Whenever we have the mechanisms of tolerance fail, we have autoimmunity. I talked about commensal bacteria symbiosis, means the normal homeostasis between immune system and normal flora of the body. And if there is no homeostasis, normal homeostasis, if in normal situation we do not have any immune response against microorganism, normal flora, which is called symbiosis. And whenever immune system reacts against normal flora or commensal microorganisms, it's called dysbiosis. We have two different terms. Dysbiosis versus symbiosis. Symbiosis means homeostatic relationship between immune system and normal flora. Dysbiosis means we do not have homeostasis. And besides, immune system should tolerate the fetus of pregnant female, and the mechanisms that control unresponsiveness of immune system, the mechanisms that control unresponsiveness of immune system to fetus of pregnant female and commensal microbes are similar to self-tolerance. So we have some mechanisms in self-tolerance that they are used to tolerate fetus, and they are used to tolerate normal flora. We have same mechanisms of controlling immune system unresponsiveness towards fetus, commensal microbes, and self. You can see some of them. What do you know about these terms? Tolerogen versus immunogen. What do you know?
Autologen reaction, no reaction or response in the immune system. Yes.
Tolerogen, no reaction in?
Yes, response immune system.
No response from immune system?
Yes.
Yes.
Immunogen reaction, stimulation reaction in the response in the immune system.
Yes. Both of them are antigens. Everything in the environment inside the body, everything is antigen. Is it clear? Everything. It's because immune system can bind to anything in the environment and inside the body. All of them are antigens. But some antigens are immunogen, but some of them are not immunogen. Immunogen means immunogeneration, production or induction of immune response, generation of immunity or immune response. So any antigen that can induce immune response is called immunogen. Like antigens of pathogenic bacteria and other pathogenic microorganisms. All of them are immunogenic. But some antigens cannot induce immune response, cannot activate immune system, like self-antigens. They are called tolerant. Okay? They are opposite. Tolerogens are indeed antigens that induce tolerance, or we do not have immune response against them. And immunogen are the antigen that induce immune response. They can active lymphocytes and other cells of the immune system. Let me ask a question. All of the immunogens are antigen. Is it correct or incorrect? All of the immunogens are antigen. Is it correct? Please. All of the immunogens are antigen. This sentence is true or not?
Correct.
What do you think?
Correct.
It's correct.
Yes.
Anything in the environment inside the body is antigen. Anything. But it doesn't mean that we will have immune response against them. We have two types of antigen. Immunogens, tolerogens. Immunogen, immunogeneration, immune response, activation of immune cells. Tolerogen, induction of tolerance, having no immune response. If an antigen to be immunogen or tolerogen, it depends on the nature of antigen as well. So, both the activity or the mechanisms of immunologic tolerance or tolerance mechanisms and the nature of antigens, both of them, affect the fate of an antigen to be tolerogen or immunogen. So, if a specific antigen to be tolerogen or immunogen, it depends on the nature of antigen and the state of immune response, the state of immune system of the person. I'll talk about.
So it doesn't depend on the variable part? It don't depend on the variable part?
About what?
Variable part of receptor. There's constant and variable.
Variable part of the receptors. When we say it depends on when we say the immunogenic to be immunogenic or tolerogenic of an antigen is dependent on the nature of antigen and the state of immune system, of course it depends on the variable region of. But we are not talking about the variable regions. We have variable regions against, for example, epitope X. We have the variable, indeed, we have the clones that contain the variable region, a specific receptor for a specific epitope. But some mechanisms of immune system try to stop the clone. Besides, the nature of antigen is important in the induction of immune system. For example, it is said that protein antigens from the lecture 1 or 2, Dr. Sharif. You should remember that protein antigens are more immunogenic compared to polysaccharide. Do you remember? Protein antigens are more immunogenic compared to polysaccharide. It's because protein antigens can activate both B cell and T cell, but polysaccharide cannot activate T cell. Why is that?
It's bounded by MSC.
MHC. Protein antigens can bind to MHC, but polysaccharide cannot bind to MHC. So only proteins activate. Besides, the diversity or variety of epitopes on the protein antigens are higher than polysaccharide. In polysaccharide, we have repetitive epitopes. For example, 1 or 2 epitopes are repetitive. But in protein, we have several epitopes. Whenever we have several epitopes, please, whenever we have several epitopes on a specific antigen, we have a stronger immune response compared to to an antigen that only have one epitope. Why it is so? What's the reason behind it?
Structure of a protein from antigen to another one.
No. Let me write it here. We have two antigen, two different antigen. One of them has, for example, 10 different epitope, and one of them has only one epitope. Which of them is more immunogenic? The first one. The first one is why? The first one is more immunogenic. Why the antigen that contains more epitope is more immunogenic? Why is it so?
Because we have more antibodies against
Why we have more antibodies against these antigens compared to this? I'm talking about the epitope. It doesn't mean the number of antigens. For example, if a cell have this antigen, another cell have this antigen, this cell have 1,000 antigen, but in 10 different epitopes. But the other one has 10,000 antigen again, but all of them are same. For example, here we have 1,000 of each epitope. It doesn't mean that the number of total antigen of these cells are different. The number of antigens are same. For example, the number of antigens are same, but the diversity of epitopes is different. Why? When we have more epitopes, more different epitopes, the antigens will be more immunogenic.
Because our body generates random antigen. That antigen may be identify this epitope, so it can.
This is antigen. Do you mean immune system?
Yes.
This is antigen. We have 10 different epitopes, one epitopes. What the number of all the total antigens?
I mean, when our body generates random antigens.
Random receptor, do you mean?
Yes, random receptor. Maybe one of these random receptor identify this as antigen.
You're talking about the availability of the clones that can recognize them. No, we have clones, enough clones inside the body can recognize all of them. There is no lackage of or shortage of clone inside the body. We have enough clone inside the body. More epitope, more clones. One epitope, one clone. If we have two epitope, we will have activation of only two clones. Whenever we have 10 epitopes, we have activation of 10. So a protein, it is called complex antigen in terms of immunology perspective. Whenever we have complex antigen, we mean an antigen which has more epitopes, complex antigen, it's more immunogenic. It's because complex antigen has different epitopes, so it can activate more clones of lymphocytes. But simple antigens that has only one or two epitopes, it can activate only one or two clones. Activation of more clones means a stronger immune response. So it's more immunogenic. More immunogenic means activation of a stronger immune response. Less immunogenic means activation of weaker immune response. So more epitopes, more clones activation, a stronger immune response means more immunogenic. Clear? So it depends on the antigen and the state of immune response, because we have some process that prevent activation of T cells or B cells. So unresponsiveness of immune system against one specific antigens depends on two sides of the battle, both antigen and immune system. We have battle between antigen and immune system. So having tolerance or having immune response, immunogenic, is dependent on both sides of the battle. Both the immune system and antigen. Both of them are effective. For example, same antigen may be administered in different ways to induce immune system. Same antigen. Antigen A, if we administer, we inject a specific antigen from, for example, from skin, it can induce immune reaction, but if we take that antigen orally, we won't have, for example, immune response. So having unresponsiveness doesn't depend only on the immune system. It depends on the antigen as well. For example, some antigen maybe for person one, for maybe immunogenic for person one, but tolerogenic for another person. So this antigen could be immunogen, but it depends on the immune system of different persons. It's because some people have autoimmunity, but some others do not have. It's because some people have allergic reaction, but some people, because immune system of the people is not same. Question, comment? دکتر محمد درسته؟ دکتر محمد جان بپرس تو رو خدا اگه سوال دارن بگن. همینو بگو. بگو تو رو خدا.
دکتر يقول الله عليكم اذا عندكم سؤال سالوا. لا بل ننصح فيه.
Please, if you have question, ask.
I'm asking.
Yes, thank you. I would be thankful if you ask question. Please ask questions whenever you do not understand the concepts. Concepts are more important. Concepts are more important than the memorizing some details. Please louder. Come here. Bodo bia. Bodo. Bodo means run. There is sit here. Please. Listen. What's your question?
تعال قدام تعال قدام. قوم قوم قوم.
What is immunogene? Immunogene is an antigen that induces immune response. Immune gene from generation, induction, production, and immune means immune response. Any antigen that is able, that can induce immune response is called immunogen. Immunogeneration, immune response generation, induction of immune response. It's opposite of tolerogene. Any antigen that cannot induce immune response, it is called tolerogene or non-immunogenic antigen. All antigens are not immunogenic. Is it clear? Yes. This is the most important concept of this section. All antigens, self, non-self, environmental, food antigens, microorganisms, all antigens are not immunogen. Is it clear? Some of them are tolerogenic or non-immunogenic, like self-antigens, like antigens of commensal or normal flora, like antigens of fetus in the pregnant women. Got it? Another question. So, as you know, everything, every action in immune system is based on a specificity. Every action, detection of a specific antigen, immune response induction, or having no immune response. So anything is based on a specificity. Whenever we talk about specificity, we talk about receptor binding. Both TCR, BCR, and innate immune cells, all of them have receptors for different epitopes or antigens. If I want to discriminate between you, I see you and I can detect the difference between you with my eyes. The eyes of immune cells are receptors. Cells can see the antigens with their receptors. Indeed, they can detect or sense them, like touching. Okay? So, if the receptors bind to a specific antigen, if the antigen is specific to the receptors, we might have immune response. Sometimes we do not have immune response. Although we have binding, we have a specific binding. When we say immunogenic or non-immunogenic, it means that we have a specific binding. As I said, we have self-reactive T cells. When I say we have self-reactive T cells, so we have self-reactive receptors that they can bind to the antigens, but they won't become activated. So tolerance mechanisms or unresponsiveness is specific, like immune response induction. For example, if we get HPV vaccine, we will have immune response against hepatitis B virus. We won't have immune response against COVID-19. This is a specificity. The clones against indeed anti-HPV clones are activated, but anti-COVID clones are not activated by the antigens of HPV. So this means a specificity. This specificity is for tolerance as well. We have a specific, when I have self-reactive clone, so I have clones that can detect my self-antigens, but they cannot induce immune reaction, although it can bind to them, although it can recognize them, to detect them. Is it clear? Both B cells and T cells. Tolerance is induced in adaptive immunity. And as you know, adaptive immune system can control innate immune system as well. It doesn't mean that innate immunity is not involved in tolerogenic or tolerance. As you saw yesterday, we have tolerogenic dendritic cell. Do you remember? In the intestine or in the skin, we have tolerogenic dendritic cell. Tolerogenic dendritic cell cannot induce immune response, although they present antigen in the cleft of HLA, but they cannot induce immune. They are tolerogenic. So the tolerance occurs in T cells and B cells, but it doesn't mean that we do not have involvement of innate immune response. As I said yesterday, whenever we have immune reaction, whenever we have immune response, we have a network of cells. Innate, adaptive, antigen presenting cells, T helper 1 or B cells or T cytotoxic or eosinophils, NK cells, a network of cells try to fight with the macroorganism. In tolerance, again, we have a network of cells. We have antigen presentation. We have tolerogenic TC4 antigen presentation. We have induction of tolerance in T and B cell. Let me skip this one. So, why knowing the mechanism of tolerance is important? Is knowing the mechanism is only for passing the exam, knowing the mechanisms and passing the exams and to be a doctor, is it enough?
No.
We can use the mechanisms as therapeutic to help us in the treatment of diseases. Recently, the immunotherapy against cancer that we have a lecture team immunology in next days. immunotherapy using of immune real cells, immune components to treatment of cancer, to treatment of autoimmunity, has evolved and has revolutionized the treatment of cancer and autoimmunity, and allergic reaction, and other diseases. It's because in recent decades, we know the mechanisms, we know the details of the mechanisms, and now can use these details, can change the details in order to manage a disease. For example, if we have a T cell that attack the skin in specific autoimmune disease, if I know the mechanism of activation of this T cell, I can use opposite action to suppress this T cell. So if I know the mechanisms, we can treat the disease more specifically and more efficiently. So all of this that I'm telling you is not only for the exam. It's important to knowing the concepts in order to help you in the treatment of the disease. دکتر محمد جان ما یک چیز بهشون بدیم جان آره یک اکتیویتی بهشون بدیم یه درگیرشن بدیم گروهی بهتره منطقه خیلی کوتاه ببینم اینجا من نمیدونم چرا اسلایدامو تغییر دادم ولی نیومده اینجا الان یه لحظه من پیدا کنم اول 3 دقیقه وقت دارم با هم صحبت بکنم 3 دقیقه antigen or peptide from antigen. So for T cell activation we need APC directly. APC should present antigen for naive T cell activation. And besides we need, let me show it with this another color. We have co-stimulator binding like B7 and CD28. So for T cell activation we need 2 signals that both of them are provided by APC. Without an APC we won't have T cell activation. Is that clear for you? Without APC, we won't have activation of any naive T cells, conventional naive T cells. Is that clear? Yes. Because only APCs can express B7 or co-stimulators. First signal and second signal. Both signals can be provided by only APCs. For example, if we have a liver cell here, do liver cell has HLA class 2?
No.
Which type of cells have MHC class 2?
Macrophage.
APCs. Macrophages, dendritic cells, B cells. You haven't reviewed your previous lectures. Only APCs have MHC class 2, but all nucleated cells like liver cells have MHC class 1, all nucleated cells. So as liver cells have MHC class 1 or HLA class 1 and epitope or antigen as well. And we have TCR here as well. If it is a CD8 positive T cell. Can this T cell become activated by a liver cell?
No.
Do not see here.
No.
If we have only T cell and liver cell,
No.
can a liver cell activate naive T cell?
No.
Naive. I'm talking about naive T cell.
No, it can't.
Why it can't?
Because it's only activated by ABC.
Why?
It's
There is no co-stimulator here. There is no B720 C28 engagement here. Without second signal, we won't have any immune response, any activation of it. It's better to say any activation of naive T cells. So for naive T cell, we need HLA, we need TCR, we need APC, we need co-stimulators. But for activation of B cell, that have BCR, B cells have BCR. Do we need APC for B cell activation? No, there is no need for HLA or antigen presentation. B cells can become activated with soluble antigen. For example, if a bacteria binds directly to a B cell can become activated. But for naive B cell, again we need two signals. First signal by BCR and second signal, as I said yesterday, with complement receptor or PRRs. Because B cells have both of them, and we had complement on the bacteria and some pump on the bacteria. Do you remember?
Yes.
So B cells with its BCR bound to the antigen on the bacteria, and plus it has PRR, pattern recognition receptors, that can bind to PAM, or it has complement receptors that can bind to the complement protein on the bacteria. So for naive B cell activation, again we need two signals. But there is no need for HLA directly. There is no need for APC. Is it clear? Please. دکتر رفتن؟ اون طبقی سوال دارم بپرسن. تو رو خدا بگید بپرسن.
طلاب عندكم سؤال؟
Doctor,
سؤال؟
can you repeat quick repeat for the different, because I'm recording now. I'm recording now.
We have no enough time. If you haven't understand, I can explain.
No,
But for recording, I cannot, because we have short time
It's a
shortage.
quick.
As I said, for B cell co-stimulation is provided by PRR binding to PAM or complement receptor binding for complement proteins on the surface of bacteria, for example. They are complement receptor, and PRRs will provide second signal. They are similar to co-stimulators of T cell. Question, comment? So, they are important, all of them are important in tolerance. For example, if a liver cell, let me go back. As I said, all nucleated cells have MHC class 1. And they are expressing self-antigen constantly. Our MHC class 1 are expressing self-antigens. Okay, is that clear? All my cells, nucleated cells, have MHC class 1 on their surface, and they are expressing my self-antigens. Is that clear? Okay. But I do not have immune reaction, T cell activation. I do not have T cell activation against my liver cell, for an example. It's because on the liver cell, as I have MHC class 1 and self-antigen and self-reactive T cell, so I have liver-reactive T cell. I have self-antigen presentation by liver cells on the cleft of HLA class 1, but there is no immune response. Why there is no immune response here?
Because
There is no co-stimulation. It's one mechanism, for example, of tolerance. Because our cells do not have, in normal condition, co-stimulators. As they do not have co-stimulators, they cannot activate naive T cells. But whenever we have, for example, bacteria or tumors or other invading microorganisms, they are engulfed or phagocytosed by phagocytic cells like dendritic cell and macrophage, and dendritic cell and macrophage have co-stimulator and MHC together. Is that clear?
Yes.
Please review the previous lectures. Do not leave them for the night before the exam. خب دکتر فکر کنم اینا نیاز به استراحت دارن.
اوكي
10 دقیقه.
10 دقائق
چند